Spread of Aspergillus flavus by Navel Orangeworm, Amyelois transitella, on Almond by Michael Zullo. Navel Orangeworm, or NOW, damage to almond is correlated with increased incidence of aflatoxin contamination caused by Aspergillus flavus. However, no reports demonstrate a causative relationship between NOW feeding and A. flavus infection. To demonstrate the potential of NOW to act as a vector of A. flavus on almond, NOW eggs were dusted with A. flavus and incubated in microchambers adjacent but not touching agar plates of almond kernels. Following egg hatch, A. flavus colonies developed on agar along trails left by NOW larvae. Almond kernels damaged with A. flavus carrying NOW showed higher incidence of A. flavus colonization and aflatoxin contamination than control treatments. Interestingly, levels of aflatoxin in NOW damaged A. flavus infected almond were significantly higher than control treatments, even in the absence of visible fungal growth. Commercial almond orchards had a relatively low level of contamination with Aspergillus section flavi in spring and early summer, and a high level during summer corresponding with the higher level of NOW infestation of the crop. Our study demonstrates that NOW is capable of vectoring A. flavus to almond, and that monitoring and sorting of almond kernels for insect damage is warranted to limit aflatoxin contamination potential, both before and after harvesting. NOW overwinters in mummy nuts left on the tree or on the ground during the winter, and its first month flight begins in mid to late March. This lepidopteran pest can complete 3 or 4 generations during the season. The highest potential for almond damage occurs when the kernel becomes accessible to the larvae emerging from hatched eggs that were laid on the hull of the almond. The larvae can chew through the soft shell or through the cracked shell and feed on the kernel, creating wounds accessible to fungal invasion. Damaged nuts are associated with increased Aspergillus flavus. The eggs are typically laid in spring and early summer on unharvested nuts, which are called mummies, from the previous season, and from late June through October on the current nut crop following hull split. Both mummy and new crop nuts may contain aflatoxigenic species, and these infected nuts may act as inoculum sources for the spread of Aspergillus species to unaffected nuts. No studies have directly determined whether NOW can play an active role in aflatoxin contamination of almond by either transporting A. flavus spores to the nuts or directly contaminating the kernel while feeding. To investigate the possible role of NOW in contaminating almond kernels, we developed in vitro methods to isolate A. flavus spores from both growth media and almond, ensuring that inoculation could only occur through larval movement and feeding. In addition, kernels damaged by NOW larvae were analyzed for their aflatoxin content to determine whether the extent of insect damage affected the levels of aflatoxin contamination. Finally, the seasonal incidence of Aspergillus section flavi was determined for almond mummies, NOW larvae, and NOW moths collected from commercial almond orchards during the season. So there are basically 5 different tests conducted here. First was the spread of A. flavus by NOW in vitro. Control treatments consisted of paper towel discs containing NOW eggs without A. flavus spores and egg-free paper towel discs dusted with A. flavus spores using a cotton swab. There was also spread of A. flavus by now on almond kernels. Almond fruit were harvested immediately following hull split, and kernels were removed from the shells. The third was incidence of Aspergillus section flavi on now moths trapped in almond orchards. Sticky traps containing 3 virgin, now female moths in mesh bags were hung 2 meters high in the tree canopy and monitored from March to October in 2 almond orchards. Also was Aspergillus section flavi prevalence in almond mummies. 500 mummies were collected from an almond orchard in Madera County. The mummies were shelled. The kernels were classified as healthy or moldy. The moldy kernels were then classified according to their macroscopic color as black, green, or white. And finally, the prevalence of Aspergillus section flavi on moths that emerged from almond mummies. So approximately 350 or 400 mummies were placed in buckets and covered with mesh with 2 millimeter square openings. The buckets were incubated at 25 degrees Celsius, and the moths were emerged and gathered on the inner surface of the mesh. So the results of these tests, the results of the spread of A. flavus by now in vitro, treatments containing now eggs and A. flavus spores showed growth of A. flavus in larval tract patterns, indicating that now larvae had picked up A. flavus spores inside the microcentrifuge tube and then deposited them on the agar surface. Following sporulization of A. flavus colonies on these plates, now larvae crawling over the colonies were observed to accumulate candia upon their setae. In contrast, no fungal growth occurred on plates containing now larvae in the absence of A. flavus, indicating that the larvae didn't harbor the A. flavus spores. There was also no fungal growth on the plates with microcentrifuge tubes containing only A. flavus spores, demonstrating spatial separation between the A. flavus spore inoculum and the medium. So on the test of spread of A. flavus to almond kernels by now, almond kernels treated with now plus A. flavus showed feeding damage in 93% of kernels. And kernels treated with now only without A. flavus showed feeding damage in 85%. Almond kernels treated with A. flavus and untreated kernels, both without showed no insect feeding damage. Visible external A. flavus growth was observed in 11% of the damaged almond kernels exposed to now plus A. flavus. No A. flavus growth was seen in damaged kernels exposed to now only or intact unrelated kernels. Aflatoxin content of now damaged almond kernels. Individual nuts from all treatments were analyzed for aflatoxin content. Of the almond kernels exposed to now plus A. flavus, 30% contained measurable amounts of aflatoxin B1. Aflatoxin was found in now plus A. flavus treated samples that did not show external A. flavus growths. The aflatoxin B1 content was positively related to the extent of now feeding damage on the almond kernels. No aflatoxins were detected in the untreated nuts. So, prevalence of Aspergillus species on now moths captured in almond orchards. In the first orchard sampled, A. flavus recovered from now moths peaked from March to the 15th of June. In the second orchard samples, there were even higher levels of Aspergillus section flavi in the moths sampled. In both orchards, the prevalence of Aspergillus increased after June 15th and peaked in mid-September. The third orchard sampled for 2 years, it showed similar results. Aspergillus species in almond mummies. The almond mummies collected in winter of 2009 from an orchard in Madera County were highly infected with Aspergillus. Surprisingly, 96% of the kernels had Aspergillus flavi. Even the kernels that didn't show any now damage were heavily infected with Aspergillus flavi, 84%. Mummies even kept in cold storage were highly infected, 98%. Aspergillus in now moss collected directly from almond mummies. Now moss that emerged from the almond mummies collected from 3 orchards located in the San Joaquin were heavily contaminated with propagules of Aspergillus. The prevalence of A. flavus and A. parasiticus was 45 to 70%. So these two charts below, what it's showing is the chart on the right, B, it shows aflatoxin B1, which level of contamination and feeding sites on the almond. So there's a correlation between the damage that the moths are doing and the aflatoxin B1 contamination. And here we see on the chart on the A, aflatoxin B1 on the right under vertical column, and you see there's dark bars and there's gray bars, and the dark bars represent those with damage and fungal growth, and the gray bars represent those without the damage and the fungal growth. And even the ones without damage had fungal growth, which is what they were saying. Yes. Okay. So now overwinters as larvae or pupa and can spend as long as 8 months in an almond mummy. These mummies in turn persist in the environment for a longer time than the harvested crop. Many mummies fall to the ground and are in contact with the soil, where they can be heavily infested or infected by Aspergillus flavus. When mummies were plated on specific media, a high incidence of Aspergillus was detected, suggesting that almond kernels serve as a good substrate for the development of Aspergillus. The high prevalence of Aspergillus detected on moths that emerge from mummies enclosed in buckets also suggests there is cross-contamination when the moths touch each other. Because the prevalence of Aspergillus on moths captured in the orchards early in March occasionally exceeded the 5% level. The increased contamination of moths late in the season may occur more because the propagules of Aspergillus are found on the crop at this time, and nuts infected earlier in the season show sporulation later in the season. The high prevalence of Aspergillus in mummies can help explain our finding that now that emerged from mummies were highly contaminated with Aspergillus. So stringent limits set for aflatoxin B1 contamination of crops, including tree nuts in the United States, is 15 parts per billion, and in the European Union it's set at 8 parts per billion. Efforts to reduce damage caused by now should include improved standard orchard sanitation practices to remove mummy nuts and other crop debris, development of new almond varieties with improved insect resistance, introduction of new insecticide chemistries to improve now control, post-harvest sorting and insect-damaged almond kernels during processing, and insect control strategies during almond storage. In conclusion, this is the first study to demonstrate that now larvae can act as a vector for the transport and introduction of A. flavus onto almond kernels. Kernels damaged by now are rendered more susceptible to A. flavus growth and A. aflatoxin contamination because insect feeding disrupts the pellicle, which functions as both a physical and biochemical barrier to fungal establishment. Increased aflatoxin levels were directly proportional to increased numbers of now feeding sites per almond due to pellicle breaches. This positive relationship demonstrated the strong association between now feeding damage and increased likelihood of aflatoxin contamination. That concludes my presentation. Thank you very much.